Welcome to Nikolai's genetics lessons, and today we are going to talk about artifacts of the Manhattan plots which we built during genome-wide association studies in order to find single nucleotide polymorphisms within our genomes. It's not compared by single genome of a single person against another person. This applies to the whole population, or at least to a certain number of people which represent population. Some group represent people with a certain, say, disease, another without disease, and we plot single nucleotide polymorphisms in order to find those which are unique to certain disease. If it is disease which is only caused by single nucleotide polymorphism, we probably would see a single dot. If we study some diseases or conditions such as obesity, we are going to see many single nucleotide polymorphisms above threshold line, because such conditions as obesity are caused by multiple factors, and many genes add to this condition. But why we see here whole lines of dots, and every dot here represents single nucleotide polymorphism. And by the way, here is across x-axis we see chromosomes number 1, 2, 3, and so on. And when we go to the very end, we see that each block becomes smaller and smaller. This is because how chromosomes are organized in karyotype. Chromosome number 1 is the biggest, number 2 next biggest, and we end with the smallest, chromosome number 22, and also we have X and Y chromosomes, which are sex chromosomes and which are not present here. So this is only for autosomes. And as you see, every what looks solid block here is actually just made by thousands and thousands of dots. There is no solid color here. It's just many thousands of dots which we perceive now as solid block. But actually every block is made just from thousands of single nucleotide polymorphisms which occur along each chromosome. So let me show you how it works. And imagine that, for example, here this line of dots here actually is compressed view of the actual chromosome and single nucleotide polymorphism that happens along this single chromosome. So let me with red dots represent here single nucleotide polymorphisms which happens about every 1,000 nucleotides. So along our genome, this is what makes me and you different from each other, because in 99.9% we are the same. So 99.9% we are the same. And these red dots, which represent SNP, single nucleotide polymorphisms, occur in about 1 out of 1,000 bases. So if we have a stretch of the chromosome and about every 1,000 base pairs, we will find single nucleotide polymorphisms. Of course, they are not spread this evenly. Sometimes they can be closer to each other, sometimes we may have a long stretch without such single nucleotide polymorphisms, but on the average, what makes us different is just 1 out of 1,000 single nucleotide polymorphisms places where we are different. Of course, this is not the only reason why we look different. For example, epigenetic changes to our genome, which represent fine-tuning switches, also affect how we look like, our health, and even identical twins with the age start looking slightly different. Not exactly like probably you cannot tell the difference when children are very small, but when they are, say, 15 years old, 25, 40, 50, it's easier and easier for us to distinguish even between identical twins because of the epigenetics. And also, there are also different factors, like we also may have deletions, inversions, insertions, and so on. So now you see these dots which makes a line actually is not coming from the single space. They actually stretch in space. But because we represent every chromosome here as a number of dots, we just compressed a single chromosome, which may be millions of nucleotides. And in Manhattan plot, we just ignore all the space between SNPs. We just perceive the space as just number of dots which goes together. So the same line can be represented by only SNPs which goes in certain order, and we can completely ignore the space between them because it is the same code in all the people. But nevertheless, these dots actually are spread on the chromosome and separated by thousands of nucleotides. And during meiosis, of course, two chromosomes, one which we got, say, from mother side, another from the father side, they go through the crossing over. So crossing over may happen here, here, in thousands of places along two chromosomes when they line up during meiosis. And that means that, let's say, we take this nucleotide here, single nucleotide polymorphisms here, and if we'll take a look, during meiosis, this crossing over may happen between these two single nucleotide polymorphisms. It also may happen here between these two single nucleotide polymorphisms. And it may happen here and here. But take a look. What are chances that crossing over is going to happen between this one and this one? And you see, chances are not very good. So we expect that these two single nucleotide polymorphisms, like these two or these two, would be, in most cases, inherited together. So we would say that they are linked. But if we compare this one and this one, you see there are more chances that crossing over is going to happen between this SNP and this one. And even if we go further upstream, the greater distance between SNPs, the more chances that crossing over is going to happen, and these two SNPs are going to be inherited, would be separated, and a new kind of hybrid chromosome is going to be produced, and these two SNPs are going to end on the different chromosomes. I mean, on the different homologous chromosomes. It's going to be still the same chromosome, just hybrid due to crossing over. Now imagine that the single nucleotide polymorphism is directly connected with certain disease. Maybe it's even causing change of the second or first position in codon and change of protein or amino acid. And let's say this amino acid would happen in active zone of the enzyme, so the whole function of enzyme can be compromised. But when we build a Manhattan plot, due to linkage between adjacent single nucleotide polymorphisms, we're not only going to pull this single nucleotide polymorphism in our plot, but also those which are adjacent. And actually, they are going to spread like this. So we are going to have a picture similar to this. So instead of, if we would spread our, chromosome, because what you see on the plot, it is condensed form, you would see our single nucleotide polymorphism of interest here on the top, and all those adjacent would also be pulled up because they are very close to SNP of our interest, and the further you go upstream or downstream, then they're going to be lower and lower because crossing over may happen between dots, between SNPs, and the greater distance, more chances that crossing over is going to happen. But why we see here straight line and not a picture like this? Imagine, in order to save a space, we compress our results, let's say twice. And then our dots are going to look like this. So the spread of dots is going to be like this. What if we compress them even twice again? And our spread of dots are going to be like this. So when we compress thousands of times, this is what we see in Manhattan plot, we actually going to get just straight line of dots. We perceive them as a straight line of dots, but actually if we stretch our chromosome, they're going to look like on this graph. But when we compress all the information, we are going to get a straight line of SNPs. But you have to understand that only the one on the top would have significance for our studies, and the rest is just probably have no effect. They just happen to be very close on the chromosome to the single nucleotide polymorphism, which cause certain disease. In only 10% of the cases, this is going to be some nucleotide polymorphism within coding sequence of the gene. And in 90% of the cases, it's going to be somewhere else, but nevertheless, it's going to be significant because it may happen in a sequence which affect how gene is expressed. Let's say in enhancer region or promoter region. So we are not even going to see any change in proteins, in protein structure. Only 10% of SNPs account for such changes. And this is all for today. Subscribe and see you in the next video. Goodbye.